Okay, so the first lecture is going to be on genetic disorders. So with that, we're going to talk about some background on DNA, molecular biology, background of genetics, and then we're going to go into different types of disorders. There's Mendelian inheritance disorders, where we talk about like Punnett squares and then different probabilities of getting a certain disorder, and then there's non-Mendelians that don't specifically apply to Mendelian genetics, but are things like chromosomal disorders where you have an extra copy of a chromosome or a lack of a copy of a chromosome. And then we'll finish up talking briefly about screen diagnostic tests, but we're not going to go into a ton of information about that. So DNA, right? Deoxyribonucleic acid. It encodes the entire genome for an organism, densely compacted into chromosomes, and it's composed of A's, G's, T's, and C's. This is a lot of basic stuff that you learn even in biology. The central dogma of biology is you go from DNA to RNA to amino acids to protein, right? Where this is the single structure. A lot of people couple the amino acids and the proteins together. And then there's also reverse transcription, which some viruses do, where it goes RNA to DNA and it reverses that, encoding things like HIV does that to incorporate its genome into host cells. You have DNA replication to replicate your DNA, transcription DNA to mRNA, and then translation is mRNA to protein. So this is the first thing that I want to emphasize here with my course specifically, is that you can see on the last slide, these are not bolded words, and in this slide there's these bolded words. Only information that is bolded will be tested on the test. So what that means is that, like, this is fodder for questions. So when you're going to study for this, I don't want to say just completely ignore everything that's not bolded, but it will directly not be tested, which is a way that I can limit some of the information and make it a little bit easier on y'all. Replication and transcription. So both occur in the nucleus, right? You have replication of your DNA. There's a whole bunch of machinery associated with that we're not going to go over. Same with transcription, where you're turning DNA into RNA. But there's these DNA-dependent DNA polymerases that replicate the DNA, and then there's DNA-dependent RNA polymerases that reverse transcription. By HIV has an RNA-dependent DNA polymerase, so it just says what the template is and then what the end product is. And that's just all to get through the process of replication and transcription. Translation occurs with a whole bunch of molecular factors that we're not going to go into. Maybe if you've taken a biochem class or a molecular biology class, you'll go into all those factors. It's very complicated, obviously, but translation occurs in the cytoplasm at the site of ribosomes, where they read this mRNA and they then synthesize the protein. And so the translation of this mRNA into proteins is where you get certain phenotypes that pop up, and that's what makes your protein structures, and then the proteins build up into the higher levels of organization that makes up a person. And mutations in these genes that occur at the RNA or DNA level is where there's a lot of problems that we're going to be talking about today. And so that's where the genetic disorders originate, because you can't express proteins in a correct manner, right? And so we'll talk about sickle cell anemia and cystic fibrosis as two examples of where genetic mutations that are inherited from the parent are the problem, and that's what causes these diseases. As far as gene expression goes, gene expression is tightly regulated and varies immensely from cell to cell. So you can imagine that if you're a neurotransmitter and you're making the pathway that synthesizes acetylcholine, which is an important neurotransmitter from neuron to neuron, you don't need to make that in a liver cell, which is aimed to detoxify different poisons, right? You need more smooth muscle, smooth ER machinery, and things like alcohol dehydrogenase to break down when you're hungover from a nice Friday night. And those processes of what is expressed, whether you're a alcohol dehydrogenase are neurotransmitters, all regulated immensely by cell signaling, transcription factors, epigenetic modifications, a whole bunch of different stuff. We're going to be talking about glycosylation changes in cancers today. So it's just a lot of regulation in biology, but we don't need to go over all the specifics right now, at least. Epigenetics is something that's really been popped up in the past 20 years, especially in lab settings and how it affects different disease progressions. And what this is the ability to turn on and off genes. So you can imagine that if you have an acylation of a gene or a histone that regulates the gene, what that means is that's a carbonyl group. Have y'all taken organic chemistry or are exposed to that a little bit? All y'all? Yeah, so we have this acylation where you have a carbonyl group. It's a carbon double bonded to an oxygen, super electronegative. It's going to pull electrons toward it. Well, if you pull electrons toward this group and you pull it away from electrons interacting with the DNA, what you're going to do is you're going to release the DNA from the histone. You're going to open up that gene and it can be more easily transcribed for expression. That differentiates from things like methylation, which does this weird thing. A lot of organic chemistry classes don't go over it, but it does this weird thing that pushes electron clouds toward it. It does all the opposite. It closes up the DNA transcription, and then you can turn off translation of the genes. But that's just ways that you can regulate things at the DNA level. You don't need to know all that. You just need to know that epigenetics are a thing, and they're a way to turn on and off genes. There's also this other thing that's really popped up in the last 15 years about microRNAs. You might also see it as small interfering RNAs, or miRNA or siRNA. All of it's the same way. It's just different regulations with dicer and all this crap that we don't have to know. But it's very complicated. At the end of the day, though, what it does is it's an RNA that binds to the mRNA, and it prevents that translation. So it's another way to regulate gene expression that's independent of transcription, right? So again, you don't need to know all those details, just that there's ways to regulate this stuff. So yeah, we're going to be talking about DNA and how it's compacted into chromosomes. And so we have a different chromosomal organization as well. We have homologous chromosomes, which are a pair of chromosomes that have corresponding DNA from each parent, right? So if you look at chromosome 1, for example, you're going to have one copy from your mother and one copy from your father, and they're going to express the same type of genes. The thing is that if your dad has brown hair and your mom has blonde hair, you're going to have two different copies. One's going to be a blonde allele, one's going to be a brown allele, and they're going to be expressed accordingly about which one's more dominant, and that's what Mendelian genetics is, right? Where you express the more dominant allele. And those two, the brown hair and the blonde hair, it's any form of a gene that occurs at a specific locus. That is your alleles. That is two alleles for the same gene. And then you have homozygous is a loci on a pair of chromosomes that have identical alleles, and heterozygous is where you have different alleles, right? So if you're homozygous for a certain gene, that means both your parents had brown hair, right? But if you're heterozygous, that's one blonde and one brown. It's just different, right? So that becomes so karyotypes are just a visualization of chromosome. It's something that molecular biologists do. Humans have 23 pairs of chromosomes, which means they have 46 total chromosomes. I think that's actually a test question, making sure you get the numbers straight. So we have 46 chromosomes total, 2 of them being sex chromosomes, the rest being non-sex chromosomes or somatic chromosomes. And when the DNA replicates, it replicates a copy of each chromosome. And if you have 2 identical copies of the chromosomes, those are sister chromatids, which is an important thing to distinguish between sister chromatids and homologous chromosomes. So these right here are homologous chromosomes because they're both chromosomes that encode the same type of genes, right? Just this chromosome might be for your mom and this chromosome might be for your dad. However, if you like zoom into this one chromosome right here, what you'll see is that there's two chromosomes actually stuck together, and those two chromosomes that are stuck together on this single little piece right here is the same exact DNA, right? And it's even stuck together. Those two things that are stuck together, that it looks like one thing, but it's actually two, those are sister chromatids. So there's actually in this picture right here, there's a tetraploidy of DNA, meaning that there are 4 copies of each allele. There is 2 copies of, like, let's say this is hair color, right? This is blonde hair and this is brown hair. You got this from your mom, you got this from your dad. What you're seeing here is that there's blonde hair, brown hair, brown hair. And so you actually have 4 different hair color alleles here. This is what occurs right before your DNA replicates. So it's not like how your body is at a natural setting state. But in a natural state, you just don't have any sister chromatids at all. All this being said, it's a lot of background. This won't be directly necessarily test as much. Just know that we have 46 total chromosomes and an resting normal cell. If you're confused about the sister chromatid stuff, don't worry about it. That's like something that would be clarified in a molecular biology course. But what I do want y'all to fundamentally understand is how to do Punnett squares. Have y'all been exposed to this in previous classes? It can get super confusing with regard to different percentages and probabilities and blah, blah, blah. I'm going to try to make it as simple as possible, but you'll need to know how to do a Punnett square for the test. So if you're not like really, really good at them, or if you want like a refresher, please email me and then we'll set something up to go over how to do the test questions at bare minimum. You can at least get those correct. So yeah, we have the genotype and the phenotype. A way that you can remember that is that genotype is what's encoded in the genes, while phenotype is what is outwardly expressed. So a really good example here is that if we look at flower color, here you can see that if you have an uppercase B, that is a purple color, while if you have a lowercase b, that's the lack of a purple color, which turns white. So if you're a homozygous dominant, all you have is purple color, right? So that's all you're going to express. If you're heterozygous, you have one purple and one white, but the dominant allele always dominates. So it's going to be a purple flower color, even though it has a recessive gene, but it just gets overexpressed as purple. But if you have a complete lack of any dominant allele, then of course it's going to turn white. So you can see here that if you cross a dominant flower with a recessive flower, all of your offspring of that next generation, if you do the Punnett square, are going to be uppercase R, lowercase r, and that always expresses as a purple flower color. However, if you cross this uppercase R with a lowercase r, with an uppercase R, with a lowercase r, you get this kind of different probability of different offspring, where some 25% will be homozygous dominant, 25% will be homozygous recessive, while 50%, these are the same genotypes, right? 50% will be heterozygous. Does that make sense? So yeah, just make sure you know how to do that. But if not, and you'll see why that's going to become important later on. Dominant traits, yeah, I think we went over all this. Yep. So what's going to happen is we're going to talk about autosomal dominant disorders, autosomal recessive disorders, and different Mendelian genetics. So what's going to be important is that for these set of disorders, autosomal dominant, that's where you have one copy of each altered gene is enough to cause a disease. So if you have that capital N, for example, that means you're going to have that disease. And we're going to talk about, looks like 5 different conditions that are examples of this. But all these are going to be autosomal dominant. So what's going to be important, or what's going to be an example test question, is that I'm going to say you have one parent that has this disease and is homozygous dominant, for example, and you have one parent that is homozygous recessive for this. What is the probability that the offspring is going to have this disease? And so you can do the Punnett square, and then you can say, oh, there's 100% probability because all of the offspring are heterozygous. This is just like this example right here, right? You're crossing capital R with lowercase r. 100% of your offspring are that heterozygous. What I'm going to do is I have practice tests for every test. For each test is going to have a practice test, and I'll give you example questions about what's to expected, and I try to make those a little bit harder than what's even on the test. That always annoys me as a student when you got some practice test and you were like, you breezed through it, you got 100% on it, and then you went into the test and you got like a C, and it was like, that was nothing like that. So make sure you know how to do the test on the practice test, and then the questions on the practice test, and if you can't, then we can talk more about how to do these problems. Now, with that being said, we're going to go over 5 examples about different autosomal dominant mutations. The first being osteogenesis imperfecta. This is where offspring have soft bones that break really easily, and you can see here that these bones are also curved. They look misformed, and this is another example of fodder for test questions. You don't need to know every picture, but if it has this like little glowy yellow halo around it, that's like fodder for the test, for being on the test. And that could get confusing as to like what the testable material is. I feel though it's like kind of a little bit obvious insofar as, what I just want you to know is that these bones are curved, and that's just a visualization of it. But if you're ever confused about, if you're looking at a picture while you're studying, you can just send me an email with a screenshot of the picture and be like, hey, what do I need to know from this picture? It's got this little halo around it, and then I'll tell you if it's not obvious. But so don't be bashful about emailing me about that kind of stuff. With that being said, though, osteogenesis imperfecta, this is an autosomal dominant mutation of this collagen gene. And so if you inherit a defective collagen gene from your parent, it's autosomal dominant, it'll express, and you'll have these conditions. Super rare. All these autosomal dominants are kind of rare conditions insofar as the majority of the even though it's a dominant allele, the majority of the population doesn't have them, because the majority of the population is healthy, just people. But there's also some consequences here that the skeletal system is very weak because it's just not formed properly. Collagen is really important for strengthening your bones. And you have this weird malformed bones where you have bowing of the legs, arms, short stature, and scoliosis. You also have these weird eyes that they look blue. That's a problem with your collagen as well. But that's just kind of a side consequence. You don't really need to know that for the test, at least. The second one's Marfan syndrome, where it's really, really tall people. There's a lot of women have this for some reason, but there's some men. And there's a lot of like NBA people because it's like tall people that have this stuff. What it results is just tall and really, really skinny people. And they have really long fingers and arms, which probably helps in basketball a good bit. This is an autosomal dominant mutation in fibrillin 1. And that's a gene that gives connective tissues their strength and elasticity. So it's up in these people. And really the only problem is that these people are tall, which isn't a super bad problem. And they have abnormal connective tissue problems. You have some circulatory issues as well as a side consequence, aortic arrhythmia. It's because your heart's got to pump so hard to get to the extremities. That's like a fun fact with a lot of people who know things about like Great Danes and dogs are so big, like they have problems living long because they have to pump so much blood to their body relative to a small dog. Same is true with humans. But they also can have spinal issues, lens dislocation, and nearsightedness. But it's another example of an autosomal dominant. Now this is a major problem at Huntington's disease, where you have sporadic movements, cognitive decline, psychiatric disorders, and that develop between the ages of 30 and 40. And what causes this is a repeat of a CA gene codon that's found within this polyglutamine gene. And so you can kind of see a picture here that this is what normal looks like, and that you only have a couple repetitions of this. But in people with Huntington's disease, they have increased amounts of repetitions of CAG, and it causes a lot of problems, especially in your brain, and it causes this neuron degeneration. And so what's going to happen is that you get brain deterioration that starts at around this 30 to 40 years old. And there's also aspiration pneumonia, problems with the lungs, because this gene, right, these genes aren't only found in one spot, right? It's your genome, so it's every single cell has the genetic capability to turn into whatever it needs to. The only thing is that it manifests themselves in your brain, but it can also pop up in like your lungs and other things. These people also, because of social problems with Huntington's disease, is that they'll have like depression and stuff like that too. So, and again, all these diseases have a lot more like symptoms than just this. This is just a simplified version to get y'all exposed to this stuff. Neurofibromatosis is a condition where you have a whole bunch of non-cancerous tumors that form all over your body, specifically in pigmented spots. And so you can see this is a really, really bad version of it. It's usually not that bad, but you can see all these tumors on this person's back, but they can pop up all over the body. And the reason why is because you have a mutation in this tumor suppressor gene, NF1 and NF2. The thing about this one is that there's usually typically a whole bunch of mutations. This is the most common one. We'll talk about tumors and cancers actually later on today, and about the relevance of tumor suppressors. It kind of sounds like what it is it suppresses tumors, it turns off tumors, but so that one's relatively obvious, but we'll talk more about cancers later on today. But the consequences are that you have tumors develop all over your body. Tumors can also develop in your eye, and so what it'll do is your eye is like this perfect little sphere that refracts light into detection machinery in the back of your eye, right? So if you have a tumor growing on it, it'll push that and just shift all the light off of those detectors in a appreciable manner, and you can also have vision problems as well. Same thing happens with your ear. Your ear needs to recognize these sound vibrations really, really sensitively, and so if you have a tumor pushing on that. So at the end of the day, these mostly appear on the skin, but they can actually appear in various spots throughout the body. And you can see some other examples here. So this is the ear, this is the eye, but they can also pop up like in your leg and other places as well. But the big picture here is that just the tumors develop. Achondroplasia. And y'all watch Game of Thrones? I stopped after season. He is really funny. Yeah, I stopped at season 1 too. When they killed off that with whoever the main dude was, I was like, okay, I'm done with this. And this is just I'll kill off like the coolest dude.
I've been doing that for the past 2, 3, 4 years. Get into season 3 and stuff.
Yeah. Well, there's so many episodes in that TV show too. It's hard to keep up. Yeah, it's something crazy, and they're all aren't they hour-long episodes? And there's a ton of episodes. It's an endeavor. I don't have the patience for it. But what's it? Tyrion? Is that Tyrion Lannister? I don't know. Whatever. He has this condition, achondroplasia, and what this results in is just short, long bones. So that specifically means is that the rest of his proportions are actually normal. Insofar as I think he has a normal sized head and stuff like that. But the thing is that these long bones, which are your femur, your tibia, I don't know, I didn't take anatomy, but these your arms and your leg bones specifically, those are significantly shortened to where you're just super short like that. And the reason why is because of this factor called a fibroblast growth factor receptor gene. That gene is really, really important it's a complicated biology, but it's important for growing those bones to a physiologically normal length. And so at the end of the day, if you don't have that, you're going to have your long bone growth interrupted. You also have these weird things where you have recurrent ear infections, breathing problems, obesity because metabolism is significantly lowered, spinal cord compression. And for this gene in particular, fun fact is that if you're homozygous dominant, it is going to be embryonic lethal, and so every single person who actually has this and is living is heterozygous for this condition, because homozygous dominant is lethal, right? And if you're it won't develop even that far. It'll be a miscarriage effectively. And I don't even know if it'll
like even
develop to where like it's so quick. There's a lot of embryonic lethal conditions in biology, especially with genetic disorders. This is a good example of them. In research, we have to like be careful that, like, if we want to study what the effect of a gene does on viral replication, for example, if we like go in and delete that gene, sometimes when you delete the gene, it'll just kill off the whole cell line, and it's just like, oh, that's important for just the cell to live and grow, you know. So it's a thing. Biology's very much an intertwined web. Okay, so that's all your autosomal dominant disorders, and next is autosomal recessive. So what you'll have, so only people that are homozygous recessive, right? Because that's the only way you can express disorder, is that you have two lowercase a's. If you're heterozygous, that dominant allele will get covered up, and you won't have these conditions, right? But if you're homozygous recessive for these things, this is when these orders, and we're going to talk about two for these set of disorders, and that is sickle cell anemia and cystic fibrosis. The first one being sickle cell anemia. So this is anemia. Do you know what that means? It's like where you don't have proper blood function, and it's going to be, and they're very painful. What'll happen is that, right, your blood circulates through your circulatory system, and what'll happen with these is that it's called sickle cell anemia because it has this sickle-shaped blood cells, and what the problem is that the spirality of these blood cells allow it to percolate through all the capillary beds and stuff like that, and move smoothly through those things. Well, this sickle shape will jam itself in these capillary beds, and it'll be really painful, especially around the joints if you have sickle cell anemia. And so that's where the pain comes from. Autosomal recessive mutations. So what this is, it's an autosomal recessive mutation in hemoglobin gene, and what it does is because you have this mutation, it'll shift this hemoglobin gene to where it exposes these hydrophobic regions, and what'll happen is these hydrophobic regions will all stick to each other, because your cell's all water, and so it's like when you spray oil into water, it looks like it stick to each other, even though it's just the water kind of pushing it all toward together. I don't know. Y'all don't need to know that.
If I'm heterozygous, I don't have it, but I can pass it.
Yeah, so it's called being a carrier. I didn't decide if I wanted to talk about that or not, but yeah, that's very much a thing that's to be considered. Yeah, so what you can do is that if you're heterozygous and then you mate with someone who's also heterozygous, you have a 25% chance of your offspring having sickle cell anemia. The thing is that it's not something that it's a very rare condition insofar as like I don't know what percentage, I need to look up how much of the population, there's people who do genetic studies of like the world where it's like you look at how many recessive genes are out there, you know, and usually these things are really low. Yeah, we're going to talk about that next slide. Yeah. You're one step ahead of me. We'll talk about that next. But yeah, what it does is it forms these like little chains and then of hemoglobin, and what it does is that pushes the cell membrane outward, and it causes this weird sickle shape. And so what you get is sickled red blood cells, and they have a decreased lower of like oxygen carrying capacity because it's your hemoglobin genes, the molecule that carries oxygen around, is messed up, and so these people also have like oxygen carrying problems. So you're not going to have a marathon runner have sickle cell anemia, right? So what you were getting at was that we have, there's this pathogen that's very, very prevalent in Africa. It's these Plasmodium pathogens that there's like 3 or 4 different types of parasites that are this, and they all cause malaria. And what these are it's these pathogens that will come in and infect red blood cells. They will replicate within red blood cells and then released. This is a very simplified version of the malaria life cycle. But what happens is you get bit by a mosquito, it deposits these pathogens and it'll replicate in your red blood cells. It also goes through your liver. But at the end of the day, it needs that red blood cells to replicate. The problem is with this red blood cell is that the sickle shape has it causes decreased binding, and it also interferes with the replication cycle of the actual parasite. And malaria isn't really a problem in the Americas or even in Europe. It's really a problem with in Africa insofar as it needs the right temperature and it needs the right temperatures to replicate, and there's ways to get rid of this pretty easily, but you need the economics to really get rid of these pathogens, these parasites. And so it's a really big problem, especially in this little belt right here. It's sub-Saharan Africa. And the thing is that it's such a problem that actually affects like survival of people in Africa. So people who are fully healthy and have regular white or red blood cells, the parasite comes in and infects them, and then a lot of people die because it's just malaria is so bad over there. If you're homozygous recessive, you have this condition really bad, and that's also going to lead to an increased mortality. But people who are heterozygous for sickle cell anemia, where they have like a stabilizing amount of sickled cells, it decreases the exposure to malaria, but it also decreases how bad your sickle cell anemia is, to where it like helps with the survivability. So it's this concept in genetics called stabilizing selection. Y'all don't need to know that. Y'all don't need to know any of this. Don't study this. Y'all don't need to know any of this for the test per se. But yeah, it's a little fun fact about sickle cell anemia is that it's weird that everywhere else in the globe, if you have really bad sickle cell anemia, it pretty much correlates directly with death, right? And increased mortality, if you want to say it lightly. But in Africa, it's like this one little exception case where you have the increased rates of sickle cell anemia actually increase your rate up to a certain point, and then you have death. So it's this like mid ground that you want to be in. There's a lot of people who work in my department from Nigeria, which is a country, I think it's like around here in Africa, where that would be wrong. I'm really bad at geography. But it's crazy. Like they come over here like year one, and then they'll like feel cold, and then they'll be like, oh crap, and then they'll have to like go to the doctor and see if they have malaria, because it'll like go dormant in you for a little bit. And they'll like the first year of them being in Shreveport, they're like concerned that they have malaria because it's just such a problem. They might have been infected before they came over here, and then usually if you're living here a while though, you get less concerned. But yeah, whatever. All right, so that's sickle cell anemia. We also have cystic fibrosis. What this is it's an autosomal recessive mutation in this cystic fibrosis transmembrane conductance regulator gene, CFTR. And what this is it's a chloride channel. So what we'll see a lot later, but the concept's still true, that these it's a chloride bicarbonate channel. What these ion channels do is they regulate the movement of ions in and out of the cell. And this one specifically releases ions out of the cell. And what's very true about water is that if you move a whole bunch of negative charges outside of the cell, these fully soluble ions are going to go outside of the cell. Well, water goes with them effectively. Is that like the water is like bound to that negative charge, that partial positive polarization of the water molecule like binds to that and will move with the water. So anytime you move ions across the cell membrane, water will move along with it. Well, your body really relies on that water to clear out secretions. So if you have a buildup of mucus and then you have this ion channel that'll move the ions across, the water of the ion and the mucus all mix together, and then it lets your mucus be really, really fluid, and your body can move that mucus really, really well. If you have a mutation in this gene, what happens is you just don't have this ion, and what happens is that the mucus actually builds up on the surface, on various surface, and most importantly, on the surface of alveoli and your airways that go to your alveoli. And what that does is it builds up a thick layer of mucus around that structure, right? And so what that means is that if you're breathing in oxygen and there's a thick layer of mucus in between you and the alveoli you're trying to get to, which right, all that red blood cells like going past, the oxygen can't go into the alveoli into the blood. And so you have a whole bunch of respiratory issues. You can't really breathe that well if you have cystic fibrosis. Additionally, these same mutations build up in the salt in that you can't dilute your salt at all on the outside of your skin, so your sweat's going to be excessively salty, whatever the hell that means. But you also, the same problems with the mucus secretions also happens on your digestive tract. Again, just like the mucus being a blockage of the airway moving into the lungs, any nutrients that you consume will go into your gut, and then it can't be absorbed into your body. And so what that means is you have a whole bunch of digestive issues as well. Normally these ions increase the movement of water. Yeah, so kind of went over that. So you have a mucus buildup of airways and digestive system. Those are the two big parts that it cause a whole bunch of problems. But obviously this is like a global effect because it's a genetic issue, right? But these things just manifest in that way. You also have recurrent bacterial infections. That's a really long story. We won't get into that. And malabsorption of nutrients. I don't know why I put that down there instead of just part of the digestive system. Whatever. Does that make sense though? So those are all of your autosomal dominant and recessive, and so these are X-linked recessive genes. So all of those are on your autosomal. All the conditions we talked about so far are on the autosomal side of things, but this is on the sex chromosomes insofar as things that determine if you're a male or a female. So males affected with these alleles will always express these because it's a mutation in the X gene, X chromosome. And so the problem is that males only have one copy of the X chromosome while women have two X's. And so if it's a recessive disorder, right? So if you're a female and you have one X chromosome that has a dominant allele and one X chromosome that has a recessive allele, the dominant allele is going to dominate, and this disease is going to be repressed. But if you're just a male, you only have one copy of the X chromosome. If that is recessive, then you have those problems, right? So there's a lot more men who have these X-linked recessive disorders. With that being said, though, it's not impossible for a woman to get two recessive X alleles, right? Where you're just homozygous for these conditions. And there are two conditions here where it's hemophilia and Duchenne's muscular dystrophy. So hemophilia is where you have a loss of the ability to form blood clots. We're going to talk about this extensively when we get to the blood clotting lectures. I don't think I deleted those, but maybe I did. I don't know. But what this is an X-linked recessive mutation in your clotting factor gene. So this is what, when you get a cut, your body comes in and seals up that wound and clots up to where you don't lose all your blood cells and all your fluids from just a simple cut, right? Well, if you have a problem with these clotting factors that you can't form that clot, you actually do have those problems where you start to bleed excessively. Bruises really happen really badly just from simple wounds because you get like a normal person, you get punched to the arm really hard or you bump on a corner or something. You just get a little bruise. It's about that big, and then because your body contains that bruise, right? That internal bleeding, but these people just can't contain it, right? And you get these really bad bruises. And then if you get cut, obviously that the same thing's going to happen just outwardly. It's not going to be contained by the skin. And so, you get excessive bleeding. You bruise easily, spontaneous bleeding in joints, and frequent nosebleeds. Your nose is a really small capillary interface. Anyone who's been in cold environments can really testify to that. So that's just a blood loss. And this last one is Duchenne's muscular dystrophy. So this is muscular dystrophy, where you have improper development of your muscles, right? And so what it is the loss of muscle control and paralysis around 6-month-old. This really affects kids. And as you can see later, you'll see that death, but usually these people die by the age of 20. So they only get through their teens. And so what happens to X-linked recessive in this muscle protein called dystrophin, it's named after the condition. And what you do is you get progressive muscle wasting. And you get muscle weakness because your muscles aren't developing to be strong. And so the way this manifests itself, and this is kind of what I'm hinting at with the glowy triangle, here what I'm trying to say is that rectangle. I said triangle. And then I caught myself, and then I heard you saying it. I was like, oh, I'll say it out loud. Whatever. All right, you get Gower signs, and what this is that like these kids normally when kids are standing up around the ages of like 5 is is that these you can usually like these kids can usually lift themselves up by their legs at that point. It might be less than 5. I think it's like between 3 and 5 or whatever. But with people with these problems, the first way that it manifests itself is that they can't lift themselves up by their legs, and they start using their arms and then their knees to help get their torso up off the ground and to where they're standing. So they can eventually get there, but it's they just don't have the proper leg muscle development to get there in a normal fashion, and so this Gower sign is like the first way that you can kind of recognize that muscular dystrophy might be a problem if a kid is consistently doing this. And it's an X-linked recessive mutation, right? So that's all of your Mendelian genetics. So again, all of those can be determined by doing Punnett squares and determining the probability that offspring have it. And you'll see at the practice test what kind of questions you're expected to know from those. Some of it's like super simple. Some of it's like, hemophilia is what kind of disease, and you just say X-linked recessive, right? But some of it's more complicated. All right. So the first non-Mendelian genetic disorder is mitochondrial inheritance. So obviously mitochondria have. So we've all learned in like microbiology classes typically that mitochondria are bacterial derived, and so they have their own DNA. Wait, what? Yep, mDNA. And the thing is your mitochondria, whenever the fertilization process occurs, the sperm is just simply pretty much just a little packet of DNA and energy. It's not anything significant. It's really the mother who carries all the important things to carry a zygote, right? A fertilized egg. The mother has all those things. So the mother will have all the mitochondria, all the ribosomes, all the endoplasmic reticulum, all those organelles that are super important. That's all coming from the mother. And what that means is that all the mitochondrial DNA that is inherited from the parents all come from the mother specifically, and they don't come from the father. And so what that means is that all of these mitochondrial diseases are inherited from the mother. So there's problems. So there's certain genes that are expressed on the mitochondria that you only get from the mitochondrial DNA. It's not actually encoded in the chromosome, which complicates this whole gene theory. But we won't go into that. So yeah, you get all these from your mother. And so the only example we're going to talk about, I'm probably not even going to test these. I just want y'all to know generally about that concept I just said, and then that there's disorders associated with mitochondrial inheritance. The only one we're going to mention is that you have this chronic progressive external ophthalmoplegia, whatever. The problem is you'll learn very quickly. I didn't learn this course from a lecture or like words. I took this course just reading a textbook, and so the pronunciation of everything is going to be rough. But I've actually hit some words this semester that I'm impressed with myself. Like earlier I hit what was it? Neurofibromatosis, and I was like, dang, I hit that one. Anyway, what this is so you'll just need to know. I may have like one practice test question on this. I don't know. But it causes. You just need to know that it's a mutation in the It's a mitochondrial DNA genetic disorder, and it's multiple mutations in this, but it causes this these droopy eyelids. That's effectively what it is, and it's really just because you have a weakness in the eye muscles themselves. Unilateral ptosis with a P in front of a T. Wait, so the P is silent. So you just say ptosis. Yeah, anytime in English when you have a P in front of a T, you don't pronounce the P.
That's why I just pronounce it how I see it.
Yeah. Well, that's why we're learning in cancer. I think we're going to briefly mention apoptosis, which spelled like apoptosis, and everyone says because it's a popping of the cells, they like apoptosis, but you're not supposed to technically pronounce that second P because it's in front of a T. So it's apoptosis. It's whatever though. I mean, y'all can pronounce it however you want. Yeah. Everyone knows what you're talking about. All right, so there's a whole bunch of diseases as well that are multifactorial disorders, and where genetics is an important component of the disease, but it is not everything. Genes play a role, but environment and lifestyle will influence whether disease develops. So a great example of this is it on here? Oh, type 2 diabetes. Yeah. So to where like if you work out, eat right, control your weight, control your cardiovascular health, people typically don't develop type 2 diabetes if you do all those things. But if you start to slip or you don't take care of those things, not everyone will develop type 2 diabetes. There's a whole bunch of obese people who don't have type 2 diabetes, but if you have a genetic propensity to type 2 diabetes and you don't do those things, in combination, you will develop type 2 diabetes. So it's, again, biology is complicated. But again, you don't need to know all these examples. We're not going to go into all of them. There's a lot of factors that even we might be talking about over the course of the next semester, right? Yeah, there's just a lot of that contribute to this class of disorders. So the last thing we're going to talk about is chromosomal disorders, where you have a break in one or more of the chromosomes followed by a rearrangement or deletion of the chromosome part. So, or you'll either unequal division during meiosis in chromosomes causing aneuploidy, which just means you have either an extra copy of the chromosome or you have a single copy of the chromosome. You're supposed to have 2 copies of every chromosome, right? But in this example here, it's a tetraploidy, so we have 3 of them, and you get an abnormal amount of DNA. And so the first example is Fragile X syndrome, where you have intellectual disabilities, these long face, autism spectrum disorders, and males are more affected. So with Fragile X syndrome, you have X chromosome malformation, it's just a chromosome disorder, and you have intellectual and developmental deficits from this. When we talk about aneuploidy, we're going to talk about 3 specific ones. This is Down syndrome, Turner syndrome, and Klinefelter syndrome. And this is where you have, again, aneuploidy is you don't have just 2 copies of each chromosome. You're either going to be trisomatic, where you have trisomy of a specific one. So Down syndrome, for example, is trisomy 21. Yeah, you got it. And we have 3 copies of the 21st chromosome. And monosomy is where you only have 1 copy, right? And so we'll see those examples here. So again, the first one's Down syndrome. Yeah, that 21st chromosome you have 3 copies of. And it causes a whole bunch of mostly intellectual problems. You have intellectual disability, stunted growth, the increased skin on the neck, and then a flattened facial profile, small ears apparently. And there's a whole bunch of like conditions associated with Down syndrome, but those are the ones I want you to know. And then it's because of trisomy 21, right? And that contrasts to Turner syndrome, where you have a monosomy of the X chromosome. So, right, this only occurs in women. This is where This is where females only have 1 copy of the X chromosome. Now, you can get away with that because it's just like a male, right? A male has just 1 X chromosome. And so you have the genetic capability to express everything off of that, but you only have one copy. You don't have the necessary amount of translation you need from those chromosomes enough to be normal, and so you develop this Turner syndrome. It's where you have webbed neck, short stature, wide spaced nipples, and difficulty getting pregnant. And again, it's a monosomatic X chromosome. And the big thing is you have amenorrhea, so you don't have periods, and it's an infernal case. But you also have a narrowed an aorta and a bicuspid aortic valve. We'll talk about the heart a lot later on. So last one's Klinefelter syndrome, and this is where you have it's right here. It's where you have trisomy of the sex chromosome. So you have two copies of the X and you have a Y, and so you're like this. So you're technically classified as a male, but that's a political debate, I think, to a certain degree that we won't get into. But so it's these males with long limbs. They're classified as male because you have a Y chromosome. Effectively, that's like the biological reasoning, at least. But you have males with long limbs, wide hips, because you have like a female type of expression of certain genes. So wide hips, low muscle mass, gynecomastia. So you develop breasts and small testicles and lower IQs typically. Yeah. So you have this is again a chromosome disorder where you have trisomy of the so all these people are going to have XXY type of sex chromosomes. And they also have delayed puberty, weakness, and difficulty in the classroom because of the lower IQ. So yeah, it's got to be hard if you have this syndrome, just both socially and developmentally, right? All right, so we're just going to briefly fly through this because just to make sure we cover everything. It's hard going over the syllabus and a lecture and 3 lectures in one class period. This is like the most compact lecture series. But yeah, you can screen for these genetic disorders with different ways. With ultrasounds, you can look at things like skeletal disorders, to where like if you start having those curved bones, you can start to see that in an ultrasound specifically, and it's a good idea to do ultrasounds, check in. You can also look at maternal serum biomarkers. They start to take the blood of the. This is a way that you can screen for like Down syndrome. Actually, is that if you have abnormal copies of the 21st chromosome after getting pregnant, then there's a potential that your child has Down syndrome, and that DNA is just leaking into the mother's blood. But you can screen for that just with serum biomarkers. There's other ways. There's other things you can look at. That's just an example. And then so the cell-free DNA is going to be found in the serum biomarkers. But there's also these other chemicals you can look at, hormones, all kinds of stuff. There you go. Chromosome 21 copies. So you can see here that you can look at osteogenesis imperfecta with these, with the ultrasounds, because this is supposed to be a straight bone. And then, so a lot of these are just, they give like face value genetic tests that you can just look at what's going on with the thing, but it doesn't confirm or deny it, because what if it's just like a weird angle of the ultrasound, or, I don't know, or like you just have a bad readout for your trisomy 21. It's not necessarily a full yes or no answer. It just warrants further testing, but these will give more. These right here, these amniocentesis and chronic villus sampling, chorionic villus sampling. These are more invasive, but they really get to have the answer before the child is born, to where you can get actual tissue samples from the fetus while it's developing. But they're more invasive to where it's like, I don't know, if it's like a decision you got to make to whether you either perform these or not. So that's genetic disorders. Any questions there? All right, so we'll take a little break.